Good morning, ladies and gentlemen. At first, I would like to thank ISMVD giving me the opportunity. It's really my privilege to be here with all of you who are pioneers in neurovascular research. Today, my research is the effects of jugular venous reflux in aging. I'm going to share with you the story of our research progress about jugular venous reflux since 2000. Like all the other clinical studies, it is beginning with certain clinical observations. After these observations, several questions have emerged. To answer these questions, we have conducted several studies, and the preliminary data leads to a hypothesis. We hypothesized that jugular venous reflux plays a role in the pathogenesis of age-related white matter changes. Then we conducted a clinical studies to support this hypothesis. At last, I will briefly talk about our future work, the animal model of jugular venous reflux. So let's begin with our clinical observations. Internal jugular vein is the main and the largest neck vein which connects to cerebral venous system. Also, it is easily examined by Doppler ultrasonography. In all our studies, we define jugular venous reflux as a retrograde flow in internal jugular vein longer than 0.5 seconds. The internal jugular vein we detected for venous reflux include J1 to J3. And we have observed two kinds of JVR. One is JVR only observed during Valsalva maneuver, and the second one is continuous JVR. The first kind of JVR, which is much more common seen than continuous JVR, is observed during Valsalva maneuver. We could see a retrograde flow in internal jugular vein after Valsalva maneuver. Valsalva maneuver and Valsalva-like conditions such as cough, straining, or chest compression will increase intrathoracic pressure, which will impair central venous return. When increased venous pressure in central vein, which is downstream to internal jugular vein, beyond the competence of jugular venous valve, we will see jugular venous reflux after Valsalva maneuver. The second kind of JVR is continuous JVR, which is a spontaneous retrograde flow in internal jugular vein at baseline. In our subjects, we have found this kind of JVR almost on the left side. And this is much more common seen in the older population. We believe that compared with Valsalva maneuver-induced JVR, this kind of JVR had higher retrograde venous pressure. And we think that the influence to the cerebral venous system in this kind of JVR is much more long-term compared with Valsalva maneuver-induced JVR. Many neurological disorders have been found associated with JVR, such as primary exertional headache, TGA, idiopathic intracranial hypertension, and of course multiple sclerosis, which include our previous reported TGA patients and transient monocular blindness patients. After these observations, we started to ask questions. Because compared with arterial system, venous system so is JVR another kind of normal variant? Does JVR have impact on cerebral circulation? To answer the first question, we thought we need the information from the IJV hemodynamic parameters registry in a large, healthy population. In this registry, we measured cross-sectional lumen and the mean velocity at middle internal jugular vein. And of course, we have detected the appearance of JVR. We prospectively collected 349 subjects, which were free of cardiovascular disease, neurological disease, pulmonary disease, and malignancy. And the age range is wide, from 16 years old to 89 years old. And then we divided our subjects into 5 groups according to their ages. The table showed that the mean velocity in right internal jugular vein and left internal jugular vein was decreasing, and the lumen was increasing significantly with aging. Do you think I need a pointer? I'm sorry. And also the frequency from patients of certain neurovascular disease. Also, we think the result implies something else. When impaired venous outflow results in venous hypertension, the upstream vein will dilate its lumen and the flow velocity will be reduced. And if the extent of venous hypertension exceeds the ability of venous dilatation to compensate, the direction of venous flow will be reversed, which is venous reflux. So our research suggests that because of these phenomena are all corresponding to the IGV hemodynamic changes with aging, so our research suggests that IGV venous outflow impedance is increasing with aging. And at this stage, we will wonder if GVR is associated with certain neurological disease with age-related cerebral circulatory insufficiency. Let's proceed to the next question. Does GVR have effects on cerebral circulation? Because the location of interjugular vein is relatively remote from cerebral venous system, so people will wonder, could the retrograde transmitted venous pressure in GVR reach the cerebral venous system? To answer the question, we use retina vessel to study, not because it is connected to cerebral venous system. Retina vessels also share many physiological and embryonic features with cerebral small vessels. Also, retinal venous dilatation is associated with downstream cerebral venous drainage impairment. So we performed the eye fundoscope photography before and during the Valsalva maneuver and graded the retinal venue. We hypothesized that there will be an increased retinal venous diameters during Valsalva maneuver, and in people with JVR, the dilated retinal venue during Valsalva maneuver will be more than people without JVR. And this result would imply that the retrograde transmitted venous pressure could reach cerebral venous system and influence cerebral venous drainage. In this study, we recruited a 30 young population. Because of that, we have found that the older population, the older subjects have more cataract problems, which will make them hard to be looked into the retina. And also, in our experiences, we have found that the older patients have more problems maintain their head position on the fundoscope camera during the whole examination procedure, especially during Valsalva maneuver. So we use young population in this study. And the table have shown that even in the young population, we have 5 right eyes and 9 left eyes, which had ungradable retina venous because of the failed fundus alignment during Valsalva maneuver. And our first finding is that retinal venous diameter is increased during Valsalva maneuver in every subject. Also, people with JVR, which were all Valsalva maneuver induced JVR, produced a greater venous dilatation during Valsalva maneuver than people without JVR. So the result giving the evidence supporting that the retrograde venous pressure by JVR could reach cerebral venous system and influence cerebral venous drainage. The next question is, will JVR influence cerebral blood flow? To answer the question, we use transcranial Doppler to detect middle cerebral artery flow velocity. Middle cerebral artery flow velocity changes are represented as cerebral blood flow changes in most cases. At first, we have to remember that cerebral perfusion pressure, which drives cerebral blood flow, is decided by artery BP and venous pressure. We perform Valsalva maneuver, transcranial Doppler, to measure the bit-to-bit ABP change and MC flow velocity changes. Normally, there are 4 phases of ABP changes in Valsalva maneuver TCD. And phase 1 to the beginning of phase 3 is during Valsalva maneuver. we can see that the changes of cerebral flow is response to ABP changes and central venous pressure changes. And this is a common used tool to evaluate cerebral autoregulation. During Valsalva maneuver, decreased ABP and increased central venous pressure will lead to a decreased cerebral perfusion pressure and consequently decreased cerebral blood flow at phase 2. And we hypothesize that in people with JVR, there will be a greater extent of venous pressure increment and could be reflected by a more decreased cerebral blood flow at phase 2. In this study, we recruited 50 middle-aged subjects, but 8 subjects had poor temporal windows, so only 42 subjects' data were analyzed. Again, the JVR detected in these subjects were all Valsalva maneuver-induced JVR. And the results show that the ABP changes in each phase were similar between JVR and non-JVR group, but we can see that the cerebral blood flow was decreasing more in JVR group compared with non-JVR. So this result show that the retrograde venous pressure by JVR could influence cerebral blood flow. After this preliminary data, adding the other existing facts, we propose a hypothesis. We think JVR is involving in the pathogenesis of age-related cerebral white matter changes. Age-related white matter changes is patchy or confluent white matter abnormalities revealed by brain image. And the most significant risk factors, of course, is aging. That's why they named the white matter change as age-related. And the prevalence increases with age, and the incidence approaches 100% by the age of 85. Its clinical significance have been certain. It is associated with dementia, impaired gait, mood disturbance, urinary incontinence. Also, it could predict small vessel stroke, dementia, and disable. However, its etiology is still unknown. Many SPECT and PEG studies have shown that decreased cerebral flow in white matter is associated with these white matter changes. So chronic cerebral hyperperfusion is thought to be the origin. This is our hypothesis. We think that jugular venous reflux is playing a role in the pathogenesis of age-related white matter changes. And the following slides, I will talk about our rationale. The autopsy study of chronic superior sagittal sinus showed that there will be fibrohyalinous thickening and stenosis of small vessel in the deep cerebral white matter. Also, there is loss of axon and myelin sheaths in perivascular region and rarefaction of the tissue. And these pathologic findings are corresponding to the pathologic finding in age-related white matter changes. And very interestingly, we have found many clinical reports about dual AV fistula in sigmoid sinus and transverse sinus. They show these patients had progressive dementia and leukoencephalopathy. And these clinical pictures could be reversed by embolization. SPECT and DWI in these patients showed hypoperfusion and ischemia in white matters. All these clinical pictures are corresponding to that in age-related white matter changes. And the presumptive mechanism is that retrograde transmitted venous pressure could decrease cerebral perfusion pressure and cerebral blood flow in white matters. So these evidence show that cerebral venous hypertension could lead to white matter abnormalities, and ischemia is the origin. In our previous report, we showed that the height of left continuous JVR in certain subjects could reach as high as the level of inferior petrous sinus and basic plexus from sigmoid sinus. This phenomenon is similar to that in dual AV fistula in sigmoid sinus. Adding our previous evidence, we have shown that there is more frequent JVR in the elderly and JVR could influence cerebral blood flow. So we thought that JVR is very possibly plays a role in the pathogenesis of age-related white matter changes. There are other supporting facts. For example, wider retinal veinal diameter has been found to be related to the progression of leukoariosis. Dilated intraparenchymal venules are related to white matter hyperintensities, and periventricular venous collagenoses is correlating with the severity of age-related white matter changes. And these venous abnormalities could be resulted from an elevated downstream venous pressure, such as in JVR. So to support this hypothesis, we conducted a clinical study. From 2008 December and 2010 February, patients consequently admitted to our hospital due to subjective memory complaints were assessed for inclusion in this study. The inclusion criteria include 55 years or older and had a CDR score, which is clinical dementia rating score, less than or equaling to 1. Because we have found that if we use a CDR score above 1, people will have problem cooperating during the Valsalva maneuver. So that's why we put it into our inclusion criteria. And the exclusion criteria were a past history of stroke, cardiovascular disease, pulmonary disease, and malignancy. We performed brain image by 1.5 telstar MRI, T1, T2 weighted, and FLAIR sequences. And then we use Shelton scale for white matter change rating. As to JVR, we graded the severity of JVR into 3 groups. People with no JVR on both sides is graded into no JVR group. And in severe JVR group, we divide them had bilateral JVR, and at least one of them is continuous JVR. And the others will be graded into mild JVR group. This grading method is based on the concept that the retrograde venous pressure is higher in bilateral JVR compared with unilateral JVR, and the influence on cerebral circulation is more severe in continuous JVR compared with JVR observed only during Valsalva maneuver. These are the characteristics of our subjects: the gender, MMSE, the cognitive score, the clinical dementia rating, and the frequencies of vascular risk factors were similar among 3 JVR graded groups. However, subjects in severe JVR groups were the youngest among these 3 groups. This is the Shelton scales for white matter changes rating. This rating method scored different brain regions separately. They scored 3 regions in periventricular area. I'm sorry, this is not working. And 4 areas in deep subcortical region, and 5 areas in basal ganglion, and 4 regions in infratentorial region. The initial analysis showed that the white matter changes in occipital, deep subcortical, thalamus, pontine, and the total infratentorial regions were associated with the JVR severity. Subjects in severe JVR had more severe white matter changes in these colder brain regions, which means that colder brain regions seem more susceptible to JVR. Also, there's a trend that the total score of the white matter changes were associated with the JVR severity. As previously mentioned, the age among these 3 JVR graded groups were different, so we did an age-adjusted analysis. And the results show that these colder brain regions, the age-related white matter changes in these colder brain regions were associated with JVR severity only in subjects older than 75 years. Also, the whole brain age-related white matter change scores were associated with the JVR severity only in people older than 75 years. In this figure, we can see that people with severe JVR had double the whole brain white matter change scores than people without JVR in people older than 75 years. So the first finding in this study showed that age-related white matter change in Quoto brain regions seem more susceptible to JVR. It might be because of that the location of the Quoto brain region is more close to internal jugular vein, which will make them more susceptible to the retrograde transmitted venous pressure from JVR. And the second finding is that the differences in total white matter change rating scores and in certain Quoto brain regions among 3 JVR graded groups was significant only in people older than 75 years, which means that there is an interaction between JVR and aging on age-related white matter changes. And the possible explanations might include that in the older subjects, they will endure a longer duration of retrograde transmitted venous hypertension by JVR. And there might be a synergistic effect with other age-related microvascular abnormalities, which include arterial problems, venous collagenoses, decreased capillary density, and amyloid angiopathy, to influence white matter perfusion and lead to white matter changes. After these results in human studies, we still need an animal model to confirm the causal relationship between JVR and brain insult and to elucidate detailed pathophysiology of JVR. We use SD rats. Compared with human, the rat drain the cerebral venous system by external jugular vein instead of internal jugular vein. And we use subclavian artery distal to VA origin and the external jugular vein anastomosis to produce jugular venous reflux. We didn't choose carotid artery, and we choose subclavian artery distal to vertebral artery because we don't want to influence cerebral arterial supply in this model. And then we use 3D time-of-flight MRA to validate the operation. Before operation, there's only arterial enhancement. And after operation, we can see that the subclavian artery, the enhancement is disappearing, which is replaced by external jugular vein and the venous sinus, which means venous reflux in these vein roots. And our successful rate of this operation is 80%. And then we would determine the JVR effect to brain at 1 day, 2 weeks, 1 month, and 3 months after the operation by 3T MRI, T2, ADC, and dynamic susceptible contrast perfusion weighted MRI. And there were 3 to 6 rats in each time point group. We also had sham control. And the region of interest are parietal cortex, hippocampus, and piriformis region. You might ask why we didn't measure the white matter in the rats. That is because the brain of the rat is so small and the white matter in the rat brain is scanty compared with human. Also, we use 3T MRI, but the resolution is still not good enough to correctly define the white matter. So it's still a problem for us to solve. This study is still underway and under analysis, but I will present some preliminary data of our ADC changes in this model. In piriformis cortex, we can see that the ADC changes is increasing at 6 hours and 2 weeks after operation. And there's a decrease in ADC changes at 3 months after operation. Also, in parietal cortex and hippocampus, there is an ADC decreasing at 3 months after operation, which means that JVR could cause vasogenic edema at acute and subacute stage and chronic ischemia at 3 months. I wish I could present the complete data in the near future. There are further questions needed to be solved. The direction of my research is mainly on providing evidences showing that there is an influence of JVR on cerebral circulation. Less attention is paid exploring the etiologies of cerebral venous outflow impairment in the elderly. But I know gladly that many of the researchers here have already had fruitful achievements on this. Besides, I think we should explore more precipitating factors associated with JVR on certain neurological disorders. For example, in my study, I have shown that age might be one of the precipitating factors associated with JVR to influence cerebral white matter perfusion. And these two directions are helpful to find potential therapy for JVR-induced neurological disorders. Thank you again, ISMVD, especially Professor Haki, and thank you all for your attention.
The floor is open to questions. Please.
Thank you for this excellent talk. It really provides, I think, a lot of integrating factor across many of the different diseases that you mentioned, and some of the commonality that might exist between these diseases, such as the gait problems you mentioned. Gait problems relating to JVR, there's gait problems relating to multiple sclerosis, there's gait problems relating to Parkinson's disease. A lot of these areas, of course, are affected by the basal ganglial changes, and of course those in turn can be affected by iron content. And so one of the things that has been proposed by Dr. Zamboni is, what's the role of iron in all of this? It didn't seem to me like you actually went and measured the iron content with your MR at this point, but I would suggest that's a possibility for you for sure to do that. But your evidence is an early part of what we're trying to do here in studying the whole venous vasculature. So some of the things I'm kind of curious about with this registry you've got is you must have all the arterial data with that as well.
Yes.
And today, later this afternoon, I'm going to show some data for several hundred cases where we're looking at the relationship between arterial flow, venous flow, and the ratios of these two, and the importance that that plays in terms of the absolute total internal jugular venous flow, not counting reflux particularly, but I'm sure it's related to it, and how these relate to the different diseases. So I think it would be very interesting if you could take your data and go back and look at these normalizations and then do that comparison and see what the flow components are, not just the reflux components associated with this. Mm-hmm. And you may find that there's a lot more association with that than just the reflux by itself. So again, I congratulate you on a very, very interesting set of work over the years.
Thank you very much.
Zivadin of Buffalo, New York. I also want to applaud you really for the breakthrough work you are doing in this area, both on human and animal side. I read your Annals Neurology 2011 papers 5 times. We looked also in healthy people. We are collecting, actually that paper is in press now in American Journal of Neuroradiology, whether there is a connection, in this case, between CCSVI and white matter lesions in quantitative terms, which means volumetric terms, in healthy people, but found only a trend. However, our groups were much younger than yours, and you are saying that really the cutoff is 75%. So my question is, do you think that your data would become stronger if they would be analyzed in volumetric terms, I mean the white matter lesions? And did you look whether there was a better association on hypointense lesions if you had pre-contrast T1 weighted scan?
I'm sorry, what scan?
Did you do in your study T1 pre-contrast scan?
Pre-contrast scan?
Yeah, pre-contrast.
Actually, in the white matter changes study, we didn't inject the contrast.
No, I'm asking, beyond just the flare, did you do the T1 weighted pre-contrast scans
Yes.
to look how many lesions are black?
A stroke, acute stroke, lacunar stroke? Does that mean?
No, I'm asking, because we see that a reflux might be more connected with those hypointense lesions on T1 weighted pre-contrast scan. So I'm asking whether you have data on that from your paper.
No, we didn't analyze that point. I know you are interested. Maybe that's iron deposition in the brain is associated with JVR. But I'm sorry, we didn't have that data. Maybe we can go back and analyze that. Thank you for your suggestion. And also, I think, we in all our human studies, my studies show that age is a very important factor. Not just jugular venous reflux, also the lumen change and the flow velocity change. So I think the age is a very important factor to look at the CCSVI and the neurological disorders. Because in normal aging, the hemodynamic will change. So it is better if we include a wide range of aging, especially in the old population.
But in that sense, we published, I showed that on Sunday, we published recently a paper in PLOS ONE and looked 252 healthy controls, but did not find, at least not significant, correlation with aging.
Yeah, I'm concerned about that.
Continue that discussion later on because we have a few more questions to go. Tim, you want to ask?
Thank you. Chief Ping, that's very good. Tim Duong from UT Health Science Center. I take particular interest and excitement about your retinal work, particularly the measure of diameter in the retinal vessels. As you probably know, the retinal and the choroid vessels all regulate very differently. And I'm wondering two questions. One, have you looked into the dynamic of the retinal vessel diameter with respect to the maneuver? One, and second, have you looked at actually laser Doppler flow in the retinal vessel and the choroid vessels? That's very easily done, associated with the disease that you're studying.
Okay, yeah, maybe in the future I will consider that. No, we
the
haven't
dynamic
done
is very
that.
interesting because the oral regulation between the retinal vessel and the choroid vessels are very different.
Okay.
Yeah, thank you.
Thank you.
Okay, let's take one last. Okay.
Okay, very, very nice study. Just an observation and a question. An emergency room complication is intra-abdominal bleeding, which increases intra-abdominal pressure. And if you have intra-abdominal pressure elevations, this is going to increase venous resistance. And there's a consistent observation when that occurs, there's a breakdown of the blood-brain barrier that occurs, and it can occur very quickly, within 2 hours, and it's completely reversible. So do you think, which really goes very nicely with this work, but have you looked at earlier time points for something like this occurring?
Yeah, we need some collaboration from GI. Are you a GI doctor?
No.
You are an emergency department doctor. Yeah, maybe we can establish some collaboration in the future.
Thank you.
Yeah, thank you for your suggestion.
Last question, please. Yes.
A small question. The confluence of sinuses is completely separate in 1% of human beings. And if you have a complete separation, I would suspect you have a reflux which goes far beyond the inferior petrosal sinus, because there is less resistance on the part of the cortical veins, and there is more reserve for venous expansions on the part of the inner cerebral veins.
Thank you.
Okay, let's thank our speaker again. I'd like to move on to the next presentation, Flow Effects and Brain Function in Aging, by my co-moderator, Richard Hodge.
Okay. Again, I'd like to thank Dr. Mark Hakey as well as the rest of the organizing committee for the invitation to come and speak here. So I'm going to be talking today about vascular and metabolic neuroimaging we've done in my group, in particular applications in healthy aging. This story is going to partly involve the imaging technologies and how we think they have a role in understanding age-related changes in physiology and subsequently in age-related neurodegenerative and other diseases. Oops, make sure that I can do the back. Okay. So we know that during normal healthy aging, there are a number of functional changes that arise. there are reductions in processing speed, degradation of executive function, the ability to filter out conflicting stimuli, as well as progressive reduction in memory function. And so these, they're inevitable, but there is significant quality of life issue, particularly given the demographic of our aging population. And we also think that these are a useful model for the deficits that are seen in a number of neurodegenerative diseases that are significant public health issues. Now, our group has invested a lot of effort in using the Stroop task as a model, given that it allows the quantitative evaluation of processing speed as well as the additional neurological burden of having to process conflicting stimuli. So in the Stroop task, subjects are asked to name the physical color of the font on a display in the MRI scanner during a functional MRI experiment. And in some cases, the letters spell out just X's. It's easy to identify the font. In other cases, there's a conflicting textual cue, like green. The word green will be written in blue letters. And our tendency to immediately read the word is very strong, and it requires some additional neuronal processing in order to suppress that urge. And we can see quite clearly that performance of that task degrades somewhat with age, and we can filter out the processing speed aspect. So if we look at the time taken for younger in blue or older subjects in green, just simply press the button when there's no conflicting textual cue. There's a little bit of a prolongation in reaction time in the older subjects. When we look at reaction times for the conflicting stimuli, these become longer for both young and old. But if we look at the kind of additional cost of the inhibition, I guess this probably works as well, better. Then there's that's also this inhibitory processing cost is significantly increased in the older individuals. So again, we and so the performance in this, one of the reasons we're interested in the Stroop task is that this type of executive function processing is known to be particularly vulnerable to deficits in O2 availability. There's a lot of literature on how people's performance in processing, in inhibitory processing, is degraded at altitudes. There's a map of Mount Everest, and as well as in flight, space flight, and human military flight, things like that. It's a big concern. When you're climbing a mountain or flying a fighter aircraft, people need to be able to focus on the appropriate stimuli, and so we think that. That's, again, it's an interesting model for some of the degradation of vascular mitochondrial function that occurs during normal aging. So we know that vascular, with advancing age, there's an increased tendency toward atherosclerosis and the attendant inflammatory processes. We see vessel wall thickening, which interferes with gas exchange and nutrient exchange in the brain, as well as increased endothelial rigidity associated with that, which is likely to lead to chronic hyperperfusion relative to younger individuals, as well as a reduced ability to regulate blood flow during increased functional loading. In addition to that, or perhaps driven partly by vascular deficits, it's increasingly appreciated that mitochondrial degeneration is an important component of changes during aging, as well as many age-related neurological disorders. So mitochondrial are extremely important in furnishing energy for neuronal tissues. As we've seen in many of the preceding talks, brain has a voracious appetite for oxygen, and these mitochondria work very hard. And they're the site of the majority of reactive oxygen species that are produced in the brain. So they play a critical role both in supplying energy, but also in the production and hopefully the regulation of these ROS, which which are damaging to cells. So mitochondria have quite a large number of unique genetic and biochemical characteristics that lead to their progressive degradation over the life cycle of an individual. And as this decoupling occurs, it's understood that there will be abnormalities in, oh, hang on, our slides are out of sync. Sorry, there will be abnormalities in oxygen consumption, decreases in ATP generation, and increases in the production of reactive oxygen species. And again, these are, in addition to their role in healthy aging, we know that the vascular deficits are at the origin of vascular dementias, as well as thought to play an important role in Alzheimer's and Parkinson's disease, which are likely to have both vascular and mitochondrial factors. So here now we arrive at the role of neuroimaging in the study of aging. So I guess when I say neuroimaging here, I'm talking about the families of techniques that are utilized in primarily in neuroscience, which would include voxel-based morphometry and cortical thickness measures that allow detection and characterization of neuronal loss with age and different disease conditions. And that's really represents a kind of an endpoint in many disease processes. There's a lot of hope with functional MRI techniques that by being able to characterize physiological mechanisms of brain activity, that we can pick up abnormal processing at an earlier stage before it actually leads to neuronal loss. And as it's commonly practiced, fMRI seeks to answer the question, is there more or less activation in a specific network of brain structures in a group affected by a particular condition? By far the predominant form of functional MRI that's been discussed in some previous talks is the BOLD method. BOLD stands for blood oxygenation level dependent contrast. It was discovered by Seiji Ogawa in the early 90s. So in this technique, as other speakers have mentioned, changes in the tissue levels of deoxygenated hemoglobin drive changes in magnetic resonance image contrast that provide a dynamic contrast mechanism that can be used to detect task activation. So the physiological basis for this is under normal resting condition, arterial blood transits through the capillaries, there's extraction of oxygen, leading to the conversion of oxy to deoxyhemoglobin, which is paramagnetic and exerts an attenuating effect on the MR signal. During increased neuronal activity, there's a flow response that tends to increase the capillary O2 saturation to accelerate the diffusion-limited oxygen delivery. And as a consequence, the exiting venous blood is more oxygenated, so the signal gets brighter. So this is again, it's really the predominant method for bold contrast, due largely to a number of advantages. It has quite good sensitivity relative to other functional, at least non-invasive contrast mechanisms. The implementation is quite straightforward, and the analysis methods are quite well established. The cons are, this is sort of what's motivated us to go further. There's largely a qualitative technique. Physiologically, it's not really specific to any one process. It represents joint changes in blood flow, oxidative metabolism, and cerebral blood volume. And this means that in cases of vascular and metabolic pathology, there may be biases that could affect comparisons of different patient control groups or different age groups. So to go further than this, in fact, this is also a quite well-established technique that's been around for some time. There's been a steady interest in arterial spin labeling, which is an inherently quantitative technique and specific to the process of cerebral blood flow. The reason why BOLD has been more widely adopted, I would say, is largely due to the lower sensitivity of ASL. So in this method, prior to imaging readout, blood magnetization in the neck area is manipulated through spatially selective radio pulses that are applied. A delay is observed here, and during that delay, the labeled blood flows into the imaging volume, creating a small flow-dependent component, which can be isolated through a subtraction, so giving you images specific to CBF, and which can be quantitatively translated into physical units with appropriate parameter estimates of other quantities. So the advantages of ASL are that it is physiologically specific to cerebral blood flow, quantitative and absolute units. The negatives are its low signal-noise ratio. The implementation of the acquisition sequence is somewhat more complicated than BOLD. It gives a lower time resolution for dynamic functional studies. And the parameters, there are quite a number of parameters that need to be selected for quantification. These need to be carefully chosen, particularly if one's looking at different populations. So this gives us kind of a family tree of fMRI methods. Which can be broadly broken down, it's necessarily simplified, but BOLD, NASL methods. In BOLD, by far the biggest application has been task activation studies, although a growing sibling of that is the application of BOLD contrast to look at resting state network connectivity. The appeal there is that, particularly in patient groups, is patients do not need to do anything, and by looking at temporally synchronous changes throughout the brain, one sees a very well conserved network of brain regions that is disrupted in many neurodegenerative conditions. In arterial spin labeling, it's also possible to do task-based functional imaging methods, but this has largely been relegated to kind of basic physiology studies, the kind of stuff that we're doing. But it's gaining increased importance for the imaging of resting cerebral blood flow to use as a factor in the interpretation, both in the interpretation of disease mechanisms and in results from functional BOLD imaging studies. So now I can talk to our aging data. So our application of these techniques has been to study a neurologically healthy cohort, including 32 younger subjects and 57 older subjects with mean ages of 24 and 63 years. We've had them undergo a number of tasks, including a CO2 challenge, as well as perform this modified Stroop task. And we'll discuss in a few slides that the CO2 challenge is used to calculate CO2-mediated cerebrovascular reactivity, an index of endothelial function, and also perform a calibrated MRI of tasks evoked changes in cerebral oxygen metabolism. The method we used is called a dual echo pseudo-continuous ASL sequence. It was provided by our collaborator J.J. Wang, now at UCLA. And this method is quite elegant in that it allows simultaneous ASL and BOLD readouts under fairly optimal conditions for both of the independent contrasts. Getting right to the functional data, these are activation maps showing group average responses to specifically the inhibition component in the Stroop task. So it would be the conflicting responses minus the easier responses without textual conflict. And so this is so what we see immediately is that there's a network of these dorsolateral regions, frontal regions here, as well as increasingly evident in the older subjects, these parietal areas associated with interference tasks and the necessity to switch the rule that they're following during this experiment. Now these are the types of maps that are generally interpreted in a functional imaging study. And it's important to emphasize that they're z-score maps of statistical parameter. As such, they don't really tell you a lot quantitatively about the relative response amplitudes between the two groups. It would appear to suggest that the older subjects have more extensive networks of parietal activation and that perhaps the peak responses are more intense. And so we, because of the dual echo sequence, we also have cerebral, so this is bold data, we also have cerebral blood flow data, and by contrast one can immediately note that the corresponding networks are somewhat less extensive, which illustrates the reduced sensitivity of the ASL method. We can continue to see the trend of slightly higher peak responses in the older subjects relative to the young, as well as slightly more extensive activation networks in these cortical regions involved in the inhibitory response, which again we've chosen because we think it's a set of functions that's particularly vulnerable to impaired oxygen availability in brain tissues. So then, to go beyond these statistical maps, we really want to know what does this imply in terms of neuronal function. So this brings us to an area where our lab has invested a lot of effort, which is known as a class of techniques known as calibrated functional MRI, where we use gas manipulations, primarily hypercapnia, mild hypercapnia, and hyperoxia, to manipulate blood oxygenation under controlled conditions so that we can calibrate particularly the bold signal. So again, it's this is a technique that's been used since the late 90s to estimate task-induced changes in cerebral O2 consumption. Until recently, these methods only provided relative fractional changes in CMRO2 during task activation, and I'll talk at the end a little bit about work we've done recently to provide absolute measures of cerebral oxygen metabolism in micromolar units using an extension of calibrated fMRI methods. So to illustrate why calibrated MRI is an interesting technique and why one has to be careful in the interpretation of bold amplitudes, I'm going to show some data that's revealing some of the pitfalls of bold. So here we have two bold response signals recorded in time in the same patch of cortex under two conditions. And one would say that the green curve shows a lot more activation than the red curve. But what's striking is that both of these signals were recorded in the same region of primary visual cortex during very intense visual stimulation. But in the red curve was acquired under conditions where there was absolutely no bold response. To understand why this is the case and to sort of spill the beans that this was a bit of a contrived situation, we show the whole picture now, which is that these red and green curves were extracted from a time interval in a longer experiment where we manipulated venous oxygenation during this period to drive it to very, very high saturation levels, so high that there's essentially no more dynamic range for bold responses, even in the presence of extremely intense visual stimulation. One striking side note is that the flow, in spite of the fact that we've driven the bold response to these sort of near maximal levels at which they no longer have any dynamic range to show an additional visually evoked response, the flow response as measured with arterial spin labeling is really not altered in the slightest. We can see sort of a shoulder here. So the manipulation used here is carbogen inhalation. Subjects are breathing 10% CO2, 90% O2, and the joint blood flow and arterial oxygenation changes are what drive these very high venous saturation levels. So we can see again that there's a ridiculous overabundance of oxygen, but the flow response in visual cortex is still happily progressing on top of this oversupply of oxygen as if it were under completely normal resting conditions. The general area of gas manipulations is something that we've explored quite widely. This slide here shows a progressive saturation of venous blood, starting with normal atmospheric composition air on the left. And you can see that different manipulations, such as breathing 100% O2, breathing 7% CO2 in air, lead to a lightening of I'll show the next slide, which is a little bit larger. A progressive loss of contrast in these, here we have air in the center of these venous structures, which culminates in the complete loss of venous contrast during the most CO2-enriched carbogen mixtures. And this is sort of at the basis of functional MRI methods. Now, this is a somewhat aggressive manipulation here. It's rather uncomfortable for subjects. So typically, calibrated MRI methods will use a biophysical model of the bold signal to produce very small manipulations, changes in bold and flow, and try and extrapolate this maximal bold signal way out here, which we refer to as M, using either very mild hypercapnia or hyperoxia, which is generally not uncomfortable for subjects. And this allows, once we've mapped out this baseline relationship between bold and flow signals, which we can do with things like a dual echo ASL sequence, we can then perform subsequent experiments with activation and detect changes and quantify changes in metabolism through shifts in the paired values of bold and flow measured during the task. To go back to our aging cohort, this is this M parameter. Again, the maximal bold signal change can be mapped throughout the brain. You see it exhibits gray-white matter contrast as well as hot spots in areas of large veins here. You see sagittal sinus, straight sinus. It's essentially these maps are related to the amount of deoxygenated hemoglobin present in parenchymal brain tissues. And we can see that there's a reduction in this value in gray matter in the older subjects. And that's an important factor in determining bold response amplitudes that needs to be taken into account when comparing data from different age groups. Again, it's also relevant that the functional responses take place against different background levels of resting cerebral blood flow, mapped here in the younger and older cohorts. There are moderate reductions here. This is well known, and it's detected in this ASL data. More striking are the changes in CO2-mediated vascular reactivity. We see again gray-white matter contrast here, strong reactivity in frontal areas, and this is substantially reduced in the older group. So if we come back to the functional responses, if we quantify these in regions of interest, we see the elderly groups have substantially larger flow responses, comparable bold responses. Applying the model to this, we can see that these are indicative, if we average through the entire inhibitory network, of increased oxidative demands in the older group. So finally, I'll close with a few words on the new technique we've developed, which we've termed Quoto, which allows absolute quantification of resting cerebral oxygen metabolism. A limitation of these, the pre-existing calibrated MRI methods is that they're sort of absolutely or fractionally quantitative, but they provide no information about baseline. So we think that this provides important new information. It's essentially based on a generalized calibration model, which kind of unifies the hypercapnic and hyperoxic approaches. And it applies exactly during our arbitrary combinations of flow and oxygenation. And if we perform independent measures, we get sets of equations that we can solve, that is, look for the intersection point, which provides a readout for both the M value and the oxygen extraction fraction at each voxel. That can be combined with arterial O2 content from end-tidal sampling, cerebral blood flow, and the measured OEF value to get oxidative metabolism in micromolar units, which have been in good agreement with PET, triple oxygen measures. and we're able to map these in the cortex to produce simultaneous maps of M, oxygen extraction fraction, resting cerebral blood flow, and resting micromolar CMRO2. So I'll conclude by again saying that BOLD is, although it's the predominant technique and it works very well, the responses can be distorted by differences in baseline physiology. fMRI calibration methods, which have been used largely to determine task-induced changes in O2 consumption, can also provide information about resting metabolism. And finally, differences in physiological baseline impact fMRI responses in aging population. And these are likely driven by vascular and mitochondrial deficits that are probably relevant in a wide variety of diseases. Thank you.
Questions?
Very nice talk. I really enjoyed it. Two quick questions. One is, have you looked at the gender difference between all the parameters you look at, particularly the M value and the CMR2? One, and second is, how do you explain the increase in CMR2 with age? I'm just curious.
So I'll answer I guess, two questions in order. The gender differences, so the resting CMRO2 methods, we only have in a small initial proof of concept cohort, so we don't have enough data on that to look at gender differences. Data that I didn't show here, which is good because I already went a bit over time, involves both the M value and also optical imaging techniques where we can quantify total hemoglobin and as well as oxygenated hemoglobin. And these definitely show the anticipated gender differences where it's known female subjects will have lower hematocrit and somewhat lower absolute hemoglobin concentrations in their blood. So that's is a kind of sanity check we've been able to replicate. The increase in CMRO2 in the older subjects also raises the need for having absolute baseline quantification, because in that data did not use the Quoto method, which gives absolute micromolar value. So we don't know if this fractional change is against an already depressed baseline, and it just, the neurons are saying, oh my God, we've got to inhibit, wake up guys, or if there actually is just much wider recruitment of neurons due to compensatory mechanisms. And we'll be able to better answer that when we establish what the resting mitochondrial or oxidative function is. Yes?
Is this on? Have you thought of doing some longer range studies of how rapidly this progresses in individuals and seeing if some oxygen concentrator therapy or something makes a difference?
Well, we're quite interested. We published the Quoto, the absolute quantification method, really at the very end of 2011, and we're quite excited to tune the technique so it can be applied in clinical populations and clinical applications. But to date, we have not done that.
Okay, let's thank our speaker one more time. With that, I'd like to turn over the chair to my co-moderator.
So I've got two mics here. Yes, so I'll introduce our next presenter, Dr. Mark Hickey.
Thank you very much, Rick, for introducing all those interesting concepts about the potential even for increased CMRO2 in elderly people. I think your comment about the baseline is also particularly interesting. So I will try to approach this from a different point of view. It's not Quoto, but similar idea of looking at the vasculature, high resolution particularly, so that we can try to study not just the small veins, but the small arteries as well, to look at issues such as overall microvascular flow and oxygen saturation. I've also added Ye-Ong Chuan onto this slide because he can't be here for tomorrow's session, so I have a few slides for him in here as well, and it fits very nicely with this talk. This is my conflict of interest statement, and of course acknowledging many of the students, postdocs, and faculty who have contributed to much of this material. So the interesting thing about MR angiography is when I first started doing MR back around 1983, one of the first challenges I was given by Neil Holland was, we have this ugly information in the images. We have these artifacts, and it looks like they're coming from the blood vessels, and they were basically like somebody moving when you took a picture in a camera. It just caused this blur across the pictures. So my first challenge in MRI was to see if I could figure out where that blurring was coming from and how to remove it. So in the early 1980s, we came up with the concept of flow compensation, which was basically kind of like a method of, in this case it's called rephasing the spins, but it would be like you kind of tracking the motion so that there appeared to be no motion associated with it. And we also used the fact that most of the arterial flow is fairly rapid, and so when it flowed into the slice that we were interested in, it had refresh magnetization associated with it. And so wonderfully, in 1985, we presented images on the pulmonary vessels, actually not the brain at that point, the pulmonary vessels, and the early images of MR, especially spin echo, where everything in the chest was basically black because it was moving. And after you applied this flow compensation, it was fantastic. Everything came to light. And really that was the beginning of studying the vessels with magnetic resonance imaging. So I want to show you here an example of two images. The first one on the left-hand side with a resolution of 250 microns in plane, and the second one with 500. And of course you can see lots of the big vessels, and by big here I mean 1 to 2 millimeter vessels at this point. Clearly in both of them, but if you look carefully, you can see, since that mouse is not working, if you look carefully, you can start to see some very tiny vessels coming to life. And I'm not sure if it's clear from you that far out in the audience, but the sharpness and edge definition of these vessels compared to these which look blurrier is much better. So if your goal is to do quantification of the presence of these vessels, their diameters, their flows, the higher the resolution, the better. The problem is, every time we go to higher resolution, it takes us longer and longer to do that. Well, luckily, when I first started, we were doing 0.5 Tesla. Now we're doing, actually, the first magnet I worked on was 0.15 Tesla. Then, of course, most of the world 10 years ago was 1.5 Tesla. Now many sites have 3 Tesla, 20 times better signal to noise, lots of opportunity to speed things up, and luckily in MR today, we're imaging extremely quickly. We have new methods called parallel imaging to speed things up. We're imaging with repeat times now instead of a second, not even 100 milliseconds. We're now imaging with repeat times of between 1 millisecond and 10 milliseconds, which gives us the opportunity to image the entire brain in 3 dimensions with really unheralded resolution up to now. So 3D imaging has proven to be very important. We use some other tricks called asymmetric echoes that can push the resolution to about 250 microns. And the interesting thing was we could do this 20 years ago. It just took way too long then, probably 20 minutes, half an hour. Today we can do this much faster. But there's a problem with time of flight. Time of flight has a dependence on that flow. So if there's a reduction in flow, or especially on the venous side, you're going to saturate the signal from those vessels and you're going to lose the ability to see them. So if you use a T1 reducing contrast agent, you can actually dramatically enhance the signal. So this is an example of that today, and you can see that pretty soon if we start increasing the resolution and contrast more, all I'm going to see by looking through the brain is pure white, because of course the brain is filled with blood vessels, right? But compared to those earlier images I showed you, which are so sparse in the vascular information they have, now you're beginning to see come to life on this, you're starting to see not just the arteries, but with the contrast agent, the veins also start lighting up, but you can see many more smaller arteries here than we could before. So it's not just the resolution, it's the contrast to noise that's critical. Well, there's something really magic here about how we do this imaging in MR. Usually, when you go to higher resolution, you lose signal to noise. And in fact, that's generally true for most big structures. But if you have a tiny vessel that's a fraction of a voxel, then if you double the resolution and therefore double the size of that vessel within that voxel, you actually increase the contrast to noise by square root 2, which means we've really failed to take advantage of just how much resolution we could use to make these images much better. So here's an example now of being able to visualize 250 micron images, even though I'm only using 500 micron resolution. So you're starting to see some very tiny vessels bifurcating off here. And that's still nice, but my real question to you is all this tissue in the brain that Rick was talking about here, that's all the vasculature that's feeding the gray matter, and white matter for that matter, where is it? Where are all those vessels? Well, it turns out if you look at an anatomy book, books by Henri Duvernoy and George Salomon, you'll find that there are spokes of 100-micron arteries running here in this whole region, and we're not even close to measuring those. So much of the disease we're talking about here and we're looking at is probably at this microvascular level, 100 microns and less. So my goal is, or one of the goals of one of my PhD students now, is to take those previous results, which are based on multiple PhD students, and see, can we now push this so that we can visualize 100-micron vessels? So this is actually an extraction from George Salomon's book. It's a beautiful sagittal image showing what the vasculature looks like down to the level of about 20 microns. And remember, the thickness of your hair is 50 microns roughly. So I'm going to focus here in this area in the thalamus, and I want you to look at these major vessels. Now on this image, these look like nice big vessels, but I'd like to tell you that this big one is a millimeter, this is half a millimeter, 250 microns, and then 100 microns, and these are getting on the order of 50 microns, these tiny vessels. And I draw your attention now to the fact that what we did here is we collected transverse data through the brain, and we reoriented it sagittally so that we could potentially see these. And lo and behold, by the way, this is a different person. Unfortunately, this is from cadaver brain study. This is a live volunteer. But you can see the general structures are pretty much the same. And now you can see these same vessels, and you're starting to see these smaller bifurcations. And although it's not visible here, if we were to go and look at individual slices, I'm clearly seeing 100-micron vessels. Now, if you were to have cerebral amyloid angiopathy, and you were starting to get iron buildup inside of the wall of that 50 or 100-micron vessel, I can see it. If you started to get thrombus in there, I can see it. If you have a stroke, even in one of those small vessels, and there's an embolus there, we will see it. So if you're an interventional radiologist and your goal today is to go and use a microcatheter and say, okay, I want to go and get rid of that embolus, well, now we can give you the complete three-dimensional guiding information to tell you exactly what vessel and where that stroke is. So the field has come very far. This particular scan, because I did whole brain coverage, actually took about 15 minutes. But again, I imagine probably in another 2 or 3 years, this will only take 5 minutes to do. So the problem, as you saw, though, is with the contrast agent, we see arteries and veins all mixed together. And that's kind of tough because the radiologist would rather visualize the arteries by themselves and the veins by themselves. So what we did is take advantage, again, of this bold concept that Rick was talking about, and the fact that veins contain deoxyhemoglobin, and deoxyhemoglobin has a different magnetic susceptibility than the surrounding tissue. So we use that information. So rather than using a T1 weighted scan, or in this case a gradient echo magnitude image at long echo time, we create this phase picture. And to our surprise, and this type of image was generated back in 1995, to our surprise you can see gray matter, white matter differences. Now why is that? That's because, well, we assumed that the iron content in the gray matter was larger than that in the white matter. Now, interestingly, we have a speaker this afternoon, James Conner, who's going to talk about his 30 years experience here, and many of his papers and many other papers really say that the amount of iron in white matter and gray matter is about the same. But that doesn't appear to be the case here, and it turns out that myelination takes the susceptibility of the white matter and actually makes it a bit more diamagnetic. So it's very interesting now. There's a group at NIH that has shown if you take transgenic mice that have demyelinated fibers, then you're going to, or axons, you're going to see them look just like gray matter. So in fact, what we thought we were doing in measuring susceptibility of iron can be modulated by the molecular structure of what we're looking at. So it may be that, in fact, James Conner is correct after all, but nevertheless, I'm actually happy that this is the case because now I can see motor cortex, for example. So if you want to do functional brain imaging, it's very easy to overlay this on the region of interest. And in fact, as I'm going to show you here, if you're doing imaging at very high field strength, like Yulin Gu showed the other day, and this is data from Yulin Gu, and one of my students went and visited him, Sam Barnes. This is just spectacular data of visualizing 1 to 200 micron medullary veins using this technique. So if, again, you want to do high-resolution fMRI, you can superimpose those results right away on an SWI image, and you can demonstrate perhaps why there's variability between people in fMRI. Well, they have a variable microvascular structure, so it may well be that that could be used to account for most variations we see in fMRI. So moving on, we apply this technique to look for iron in other forms, sometimes for stroke. So if there's extravasated blood, you would expect to see something. Well, here's a patient with stroke. We can't really see very much here. T1, we see nothing. SWI magnitude, we see nothing. But on the phase image, you see tiny amounts of iron. This technique is sensitive to nanomolars of iron. So you can really see things that are changing really down at the cellular level here. So here's an example of the stroke that's visualized with this technique that you just can't see under normal circumstances. Now, how else can we use this sensitivity to iron? Well, I'm going to go back to this SWI image, which is, it's a pretty picture, but it's not really quantitative. If the veins go dark, and I've lost my coffee, but I promise you I was drinking coffee just before my talk here. If the veins go dark here, it is an indication that you've had loss of blood flow there, because if the CMRO2 is the same, you're eating up a lot of oxygen, you're going to have much more deoxyhemoglobin. And I'll show you that in a minute. Here, this is a quantitative susceptibility map of the veins in this case. And so this is directly related to oxygen saturation. So if we can push this to a little bit higher resolution, I could put a cursor on here, and it's going to tell you that that thalamostriate vessel has an oxygen saturation of 68 plus or minus 2%, or 50 plus or minus 2%. Obviously, 70 is what you want, but I think we're not far from being able to do what Rick was talking about here, was having this baseline study to know exactly what the oxygenation level is. Now, people might argue that, okay, you have this vein, that doesn't tell you what the oxygen saturation is in the local tissue. Well, sure it does. On the average, it does. I don't need to look at the capillaries to say what the oxygen saturation is, as long as I have the vessel that's feeding that territory. So we use this concept to study 10 stroke cases where we imaged them the day of the stroke, 2 weeks later, 2 months later. And we measured the phase changes and plotted this versus the NIH stroke scale. And if the oxygen saturation went down, the patient got worse. If the oxygen saturation goes up, the outcome for the patient is better. So this is only 10 cases, but the potential to use this to monitor what is happening in brain function is very clear. So finally, moving on towards the applications in fMRI, if we use these really high-resolution scans, it's possible to also do functional brain imaging, but it's very difficult because the signal-to-noise is really not very good. So I want to show you an example between what's commonly done, to my surprise, in most research facilities, is what was developed almost 20 years ago, echoplanar imaging, very low resolution, 3-4 millimeters on each side, and you get this wonderful motor activation. If we use 2 millimeter isotropic using conventional gradient echo imaging, so I can do it fairly quickly, I get much better localization. Now, how can this turn into this? It's just simple blurring, and it's the method of EPI. Did EPI do a good job here? Absolutely not. This is the localization of the motor cortex. Now let's push that down to 1 cubic millimeter resolution. And here is an example of the activation associated with the neighboring tissue of that Peel vein. That Peel vein has changed its oxygen saturation. Actually, this was first published back in 1995. It's just that it took too long to do it then, and it was done at 1.5 Tesla. So the ability to really study the function in the microvasculature is there today. So I'm pretending to drink my coffee now. So if you drink 2 cups of coffee, 200 milligrams of caffeine, you're going to have a systemic vasoconstriction effect and reduce blood flow to the brain. So if you have a stroke, I would not recommend that you drink a lot of coffee. And that reduced blood flow to the brain is going to increase the deoxyhemoglobin. And so what's going to happen is, instead of seeing all these nice dark vessels here, you notice that they get even darker. You see, now you can see these tiny little veins here right at the tip, much better than you could before, because you have this systemic reduced blood flow to the brain. And when you see that effect in someone who has stroke, for example, if this part of the brain became very dark, you would be able to conclude that that was a stroke. And you can compare this directly with diffusion-weighted imaging, and you get excellent matching of that. So we can take that caffeine data, and we can apply this, what we call the SWIM, or quantitative magnetic susceptibility mapping, and you can look at the image pre-caffeine and post-caffeine, and you can see the septal veins here, for example, showing much better. That's because the susceptibility went up in those veins, and we can now directly show the change in susceptibility. Well, that's a functional brain imaging experiment. So the final thing we want to show here is that we don't always want to use a contrast agent. We'd like to be able to get this information just by using conventional imaging. So what we're going to do is marry SWI and MRA in a single interleave scan where we collect, and I'm sorry for the technical detail here, but we do a flow compensation, we collect 2 echoes that are fully flow compensated, we do a second RF pulse, we do flow compensation again, but this time we apply a bipolar pulse to dephase it. The people who do DTI are going to think of it as a bipolar pulse for creating diffusion effects, but in fact, for bulk material, it's a dephasing effect. So here we can create a dephased image. Oops. So here's an example of an image where we have just done conventional MRA, and here's the image after we've done the dephasing associated with it, and then here's the subtracted image. And it's starting to look a lot like that contrast image. Maybe not quite as many vessels, but you can see some of the veins in here, and you can see the arteries, and you can see many of these smaller arteries here as well. So just to show you what that looks like, here's the contrast-enhanced images. Here's the SWI with veins only, and here's the dephasing with veins and arteries. So now by putting all 3 of these together, you can get a complete system of arteries and veins separated from each other. So the culminating point of Ye-Yang Chuan's talk that was supposed to take place tomorrow was he's now started processing that data by using some special linear processing and nonlinear processing, and his goal is to try to get rid of the veins. And in this nonlinear processing, he can take these structures here that are not in the original MRA, i.e. no vein, but are in the subtracted image, and now he can suppress them. So it's another nice way to use this. And the beauty of this is they're all collected with the same resolution, they're all collected at the same time, and so you have a complete vascular study here. So in conclusion, it's now possible for us to really investigate down to a level of at least 250 microns the vascular system without registration artifacts. This is still taking about 8 minutes to acquire this data, but I'm sure we'll do better. And in conclusion, I think with the advent of high field imaging and parallel imaging, we'll be able to do this more on a clinical basis. We'll be able to look at modeling the vascular system so that we can predict or we can see what we expect to see from activated to resting state in functional imaging. And then when resolution becomes high enough that individual local draining veins can be seen, such as the Peel veins that Yulin Gu showed, you can be looking at functional brain imaging, which Dr. Ugerbil has much experience with, at high resolution here at fractions of a cubic millimeter. And finally, I think these techniques should be used for clinical imaging to study things like multiple sclerosis, Parkinson's disease, aging, and I think it's a very exciting time for all of us here to have access to technology like this. Thank you very much.
Okay, the floor is now open for questions.
That's good. That gets us on time.
Okay.
I have a question. Mark, so you're really aiming for very high resolution imaging in the human brain, and you mentioned 250 micron. So is this the target that we have to aim for right now? Is this what is feasible, and let's say on a clinical platform? Is this our aim, essentially, what I'm trying to establish?
Well, I wasn't quite clear enough, I guess. 250 microns you can do today on any clinical scanner using this technique. The goal is, even if I can only image with 250 microns, that I want to be able to visualize 100 micron vessels. And I think, practically speaking, if you use a contrast agent, and now we're showing even without a contrast agent, that you have the potential to even visualize the 100 micron vessels. You can't actually measure them, of course, but they have so much signal in them that, in fact, they become visible with 250 microns. So I think a rule of thumb here is you can usually see a vessel that's about half the size of your resolution. So this is a pretty exciting time frame for us to be looking at conventional acquisitions in high resolution. Thank you. Thanks, Rick.
Okay, let's thank our speaker again, and we'll... It's now my pleasure to introduce my co-moderator, Camille Ugerbil.
Thank you. Thank you very much for the invitation to present this talk here. And could I have my slides on the screen? Or do I do something from here? Ah, great. Okay, I'm going to be talking about neurovascular coupling and cerebrovascular organization, and these are my conflict of interest statements. I do get consulting fees and research support from industry. And the work that I'm going to be presenting comes from mostly, vast majority of it is going to, comes from the Center for Magnetic Resonance Research, University of Minnesota, from the laboratory that I direct. I will show some results also from the Max Planck Institute lab where I spent some time for a few years. I'm going to be talking primarily about functional imaging and also the role of vasculature and vascular control in functional imaging. This is from our very first work together in collaboration with Seiji Ogawa from Bell Labs, introducing functional imaging, showing that you can actually map signal intensity changes with stimulus in the brain. You've already heard quite a bit about the mechanisms underlying functional imaging. And in functional imaging, in these, especially in these bold or blood oxygen level dependent contrast images, we are looking at, in principle, deoxyhemoglobin content in imaging voxels. So when deoxyhemoglobin is present, you have magnetic field inhomogeneities around blood vessels that contain that deoxyhemoglobin, and these inhomogeneities are mapped in a very simple gradient or spin echo weighted image. And in brain functional imaging, we are looking at actually changes in the oxygen content or deoxyhemoglobin content in those vessels coupled to neuronal activity. And that change comes in because the fact that increased neuronal activity ends up in an increase in regional blood flow, and the regional aspect of it is extremely important. So there's a very unique spatial coupling between neuronal activity and the blood flow changes in the brain. And however, this results in a lower deoxyhemoglobin content because the CMRO2 does not increase commensurately with the blood flow. And this regional aspect is really quite important, and it has been a focus of our studies for many years now, because of the fact that from the very beginning of functional imaging, we were interested not just staying with functional images like this, where you're looking at millimeter or supramillimeter resolution organizations in the brain, such as the primary visual areas shown over here, and the lateral geniculate nucleus, which is the very first input stage of visual input. But we wanted to go from images like this to cortical columns, which are images like this, and being able to differentiate function across cortical layers as well. I want to talk about the vascular coupling information that is needed to go from here to here. But before I do so, let me just give you an update on where we are with such cortical column and layer imaging. The example that I showed in the previous image is actually orientation columns in the primary visual cortex. In the primary visual cortex, the output from the eyes is segregated in the lateral geniculate nucleus and then ends up in segregated neuronal populations, activating segregated neuronal populations in the primary visual cortex V1. And superimposed on these segregated populations from one eye versus the other eye, which are the ocular dominance columns, you have the orientation-sensitive populations, where the orientation is encoded in 360-degree representation around singularities called pinwheels. In these cases, the subject may be looking at, or the animal would be looking at grids like this that are moving up and down and changing orientation. This is actually a movie, but unfortunately it's not running in this computer. And the ocular dominance columns themselves, they look like fingerprints, and their boundaries are these black lines over here. So essentially this is a superimposition of two columnar structures in the early visual areas. So when you remove the orientation domain structure, these are the ocular dominance column structures seen that would be seen on the surface in the primary visual area. There are many examples of columnar structures, just a few other examples. For example, the whisker barrel cortex, very well studied in the rat brain. You have a one-to-one correspondence between each whisker in the rat and in the brainstem, thalamus, and the somatosensory cortex. So each of these areas, Each one controls a single whisker, and they are the dimensions of these columnar structures. And this is actually a stained map of those organizations over here shown in the postmortem brain. There are even evidence for cognitive functions organized in columnar structures. This is from a review by Tanaka showing that in the monkey brain studied with electrophysiology and optical imaging, columnar structures like this exist for recognition of faces and objects, etc. So this is this kind of structure is likely to be a principal organization mechanism in the brain, and it has been our intent to get to that level of functional imaging from the very early on when functional imaging came into existence. And what I can report today is that this is feasible. This has been done in the human brain, and of course it has been done in the animal brain for a while, but we were able to achieve this in the human brain. And this is from our paper published in 2007 by Isa Yacoub, Amir Shimuel, et al., and where we are looking at a single slice in the human primary visual area V1, shown over here. This is actually a unique individual in the sense that he was chosen for the fact that he has a flat cortex over here. So we can look at this flat cortex and we can see the ODC, or the ocular dominance columns, very nicely, and in this flat cortex we expect them to be running perpendicular to the interhemispheric fissure right over here, and we expect them to have this interdigitization as shown over here. And these are extremely reproducible images. I won't go into detail showing them to you because they're published and you can look at them in these publications. But at the same time, we were able to image orientation domains as well in the human visual cortex, and as shown over here. And as I've shown in that cartoon, we have now the boundaries of the ocular dominance columns in these right over here with these black lines, and the orientation sensitivities are shown superimposed in the human visual cortex. And this is a paper that was published in 2008. So with that effort, we were able to bring functional imaging to the level of columnar resolution in the human brain. So what was the territory of monkey or animal model imaging in general, for example, as shown over here, was we are able to do it in the human brain at the present time. I should say, of course, this optical imaging study is invasive. You have to expose the brain to get this high-resolution imaging, and you can also study it only on the surface of the brain because of the fact that the light doesn't penetrate more than approximately 1 millimeter or so into the tissue, whereas the fMRI studies can be performed in principle in the whole brain, but there are, of course, challenges of doing such high-resolution imaging in the whole brain like that, and these are challenges that we are tackling right now and we will be dealing with in few years to come still. But those studies on orientation domains and ocular dominance columns imaging were essentially introductions into this field, looking at organizations that we knew should exist in the human brain based on animal model studies, and of course ocular dominance columns can be imaged in the postmortem brain as well because of the fact that they are hardwired structures. However, we have been able to recently extend and go beyond that and go into structures that are not so well known, and this is a case in collaboration with our colleagues in Maastricht showing the orientation axis of motion selective features in human area MT. So if you look at the coronal structure in the human brain, this is the primary visual area, V1, and ultimately that information projects into much higher order areas, and in this case this is MT right over here. There are two of them bilateral, but we're going to be looking at only one side, and in that MT there are organizations that are sensitive to direction of motion. It is kind of like the orientation domains, but somewhat more detailed, more enhanced, let's say, processing of information, and they can tell, for example, motion going one way, this way, or the other way in the MT area. And these are now not a single slice studies, they are restricted volume, but three-dimensional volumetric imaging. So we can look at them on the cortical surface as well as a function of depth in the cortical thickness. And we analyzed our data in 3 layers, and we are looking at a projection of those 3 layers onto a two-dimensional surface. So they're overlapping because of the curvature here, but then you can flatten them and represent them in a flat fashion. So these are 3 layers in the MT area showing axis of motion-sensitive columns, and as a function of the cortical depth over here, as shown over in this section over here. So we have there are other examples of this. We have image columns in the parietal cortex, and we have also imaged layer sensitivity in the primary visual cortex. But with these studies, essentially, we have been able to get to a scale of brain in terms of function, which is not exactly at the synaptic cleft level, not exactly at the individual neuron level, but on the other hand, fMRI today can image at the level of cortical layers. There are, as you know, 6 neuronal layers that have been identified between white matter and the PL surface, and they do have different circuitry and different functions, and we can distinguish them. We can image at the level of cortical columns, and of course at the level of macroscopic circuitry. This has been the scale of fMRI for many years already. But to get to that level, we had to do quite a lot of fundamental studies, and in principle, because of the fact that in functional imaging, we are looking at a mediation of signals between neuronal activity and what we detect through the vasculature, both the arterial system and the venous system. Venous system because the signals that we detect come from capillaries and the veins. Arterial system because the flow increases are regulated primarily in the arterial system. So one of the very first questions we had to ask was, is cerebral physiology compatible with MR imaging of functional activity at the columnar level? As you've seen, this is the case. The answer is yes, but I want to show the study that we have done many years ago leading to that. And this is a question of cerebral blood flow and neurovascular coupling. The reason we are asking that question is if, for example, looking at something in a cartoon like this, we're looking at a columnar structure like that, and we are interested in seeing, detecting that with functional imaging. You can actually design a paradigm where the activity of the neuronal population here is increased. However, there's a blood flow that comes as a result of that. But if the blood flow supply to this area is controlled over here, if the tap is over here, let's say, then you're going to end up with a flow increase like that. And in this case, the oxyhemoglobin changes will be in this large territory. And because of this increased perfusion area that supersedes the actual spatial extent of neuronal activity, we will not be able to map this specifically. So this is a question that we had to address, and this was done in a paper that was published in 2001 by Tim Duong and Sanji Kim, and in fact Tim is going to be talking in this conference later today. I regard this as a very important paper because of the fact that it actually settled important controversies at the time about the regulation of blood flow in the brain. The study was performed in the cat cortex in a single slice study, and we are looking at, again, the orientation domains that I showed you in the human primary cortex. But of course, these studies came before the human effort and laid the foundation for the human work. So when you look at a grid like this moving up and down and changing orientation, but let's, in this case, a single orientation, we were able to get images of orientation domains. But these are not bold images. You have heard already that you can map blood flow directly, perfusion directly with magnetic resonance techniques. And these are essentially, not essentially, these are CBF-based or blood flow-based functional images. And with the blood flow-based functional images obtained in this case at 9.4 Tesla, we were able to see these orientation domains. And this is not bold, again, I want to emphasize, and it's a question we are looking at blood flow changes coupled to these orientation specific neuronal populations. When you look at orthogonal orientations, 45 versus 135, you can see that you get these complementary activation maps. And because these CBF changes can be mapped quantitatively, you can actually look at what are the extent of the changes in these activated areas. And what you see over here is that you get something like 60% blood flow change in the activated column and approximately 20% blood flow change around it. So there is in fact a big difference between, even at the column level, between the blood flow changes that take place in the columnar organizations that are processing the activation versus the surrounding ones. So there is enough contrast in the CBF signals to map that. The implication of that is CBF is regulated at the level of cortical columns. And this was not quite known at the time. And in fact, optical imaging literature had suggested, as expressed in this sentence here, that columnar specificity of flow regulation does not exist. And this was expressed by this concept here that brain waters the entire garden for the sake of a thirsty flower. But this work by Tim Duong and Sanjay Kim, etc., coming from our laboratory, showed that that is not quite correct. In fact, what happens is the brain really waters the thirsty flower while it sprinkles generously around it. So the idea then that this work has introduced is that there must be flow regulation at the level of capillaries, not only arterioles or major arteries, but even at the level of capillaries. And there has been some studies showing that there could be regulatory structures present in capillaries that can be responsible for that flow regulation. So CBF, fMRI can yield functional maps at columnar resolution, but on the other hand, as you have heard already today in the previous lecture, that CBF does not have enough contrast really to be able to do high-resolution imaging in animal or in the human brain. And as such, we had to pursue and go back to bold imaging, but these CBF studies laid the foundation of searching for those bold signals. And we had to then deal with the Venous problem, given the fact that physiology was collaborating with us. I won't go too much into detail about all the different implications, mechanistic implications of what veins and how they contribute to functional imaging. Suffice it to say that there was quite a lot of development that enabled us to get to that column level imaging in the human cortex, and this involved instrumentation, ultra high fields or 7 Tesla and higher magnetic fields, which we introduced in 1999. This is the very first 7 Tesla magnet in the world going into our center, and there has been technologies developed in many other laboratories, such as parallel imaging, as well as in ours, that have also been extremely important to be able to get to that level. So incessant discoveries and improvements in methods of image acquisition and reconstruction, this has been a very big part of functional imaging to get to that column levels. And finally, rigorous understanding of neurovascular coupling, and both simulations and animal model studies, as you have seen already. I want to show you one more animal model study where we've taken the perfusion studies, or the CBF changes that I showed you in the animal, in the cat cortex associated with columns, to a new level which may be more interesting to this audience, because we then went forward. This is work by Pat Boland and Noam Harel. Wanted to image actually the vessels as well as functional activity and correlate them. So we are not at a point where we can actually image capillaries, but on the other hand, we can see many of the intravascular structures or vessels at high fields in the animal brain. So this is a slice in the cat cortex where you see dark and light points over here. You can see them, and as you've heard from Dr. Hake's talk before, that they correspond to veins and arteries, but these are intracortical vessels. So we're looking at an image slice like this, and you can see vessels like that running into the cortex. This is a vein because it contains deoxyhemoglobin and has a different susceptibility inside versus outside, and this is the source of functional imaging signals. On the other hand, you have these bright signals that are coming in, and these are the arteries in the cortex coming from the fact that these are time-of-flight effects due to inflow of fresh spins into that slice. You can, these are high-resolution images, of course, and you can see them, you can distinguish them in these images directly because of the susceptibility difference in the veins versus the arteries. Arteries are bright. They have virtually same T2 star and T2 as the tissue, but on the other hand, veins, because of the deoxyhemoglobin content at these very high fields, have extremely short T2 and T2 star. And then you can also add contrast agents. So what I had shown you was without contrast agent. You can also add contrast agent, which then affects all the vessels. So here then everything is starting to appear dark, and then subsequently you can then see all this vasculature over here and in multiple slices into the cat cortex. You can look at them in a, hopefully this will run. So not only the PL vessels, which are showing up bright, but intracortical vasculature is now can be imaged at these high-resolution images as shown over here. And of course, the kind of signal-to-noise and resolution that is feasible in the animal models at high fields like 9.4 Tesla is not quite available in the human brain, but these are the kinds of targets that we aim to go to at human brain, as I will show you. You can, in an image like this, you can color code the vessels. So here you see now the arteries and the veins, intracortical arteries and the veins, color coded blue and red, and then we can actually look at them in terms of their relationship to functional activity, which is shown over here. And this is, in fact, a movie looking at, as a function of cortical depth, these vessels and the functional activity that goes with them. And what is interesting is essentially it confirms the study that I talked about that Tim Duong had carried out in CMRR, namely at high fields like this, you have capillary contributions as a result of which you see diffuse activation, and the activation is not just associated with these relatively large intracortical vessels. So where can we go then with respect to human efforts with respect to imaging vessels and activity like that? And here high fields are very helpful, and as I have mentioned that high fields are very important in our lab and has been part of our technology development to get to that ocular dominance or columnar level imaging. And there are several advantages here. You have long T1s for improved contrast, improved time of flight contrast. You have larger magnetization transfer for background suppression when you want to see arterial vessel. And you have high signal-to-noise ratio so that you can get to high-resolution imaging. However, everything is not that easy at, and then you have higher effects for susceptibility differences for imaging of the veins. On the other hand, there are limitations. Power deposition is a big limitation, and at the same time, B1 inhomogeneities are a big problem as well. And these are technical challenges that we have to deal with when we are doing human imaging at ultra-high fields. But I want to share with you just a few slides, some recent results of 7 Tesla imaging and 9.4 Tesla imaging of blood vessels in the human brain. This is an effort by Sébastien Schmitter and Pierre-François Van de Mortel from our lab showing time-of-flight angiography. I want to emphasize this is not a contrast agent. There is no contrast agent here. This is time-of-flight. And it actually already looks quite good as shown over here. But there are imperfections because of the fact that the RF field, the B1 field, is very non-uniform at 7 Tesla. By the way, it's not extremely uniform at 3 Tesla either, but these non-uniformities become much more severe when you go to high fields like 7 Tesla. So the transmit field is very high in the center and weak in the periphery. And those things have to be taken into account. That's quite a technological development. It involves multi-channel transmit and techniques, B1 solutions, that actually control the transmit field. But you can see that when you improve when you use those techniques, which we call in this case phase B1 shimming, you can actually visualize the vessels that are absent over here much nicer, and you can get time of flight angiography without contrast at this kind of resolution. These are 0.5 millimeter isotropic studies. And as I had mentioned, there are a lot of other improvements you have to deal with at high fields because of power deposition limitations. For example, if we take time of flight measurements, and because of power limitations, we skip saturation and MT pulses to improve the contrast. You do get angiography results. They look pretty good. But on the other hand, you can improve them substantially by using these pulses. You run into power limitations, and then you have to solve those by then dealing with the RF pulses and the MT in more intelligent ways than they are done at 3 Tesla. So if you compare the 7 Tesla versus 3 Tesla images, we did not do this comparison directly ourselves, but we asked for some angiography results from Siemens that they had considered very good, and they've supplied this work by University of Utah. And you can see that at 7 Tesla you can actually see much more vessels, smaller vessels in this area than at the 3 Tesla. And this is expected because of the higher signal to noise as well as the contrast improvements that we expect. And the 7 Tesla images, I have to say, these are our first attempts, and probably improvements because of coils, etc., are still feasible at high fields, whereas 3 Tesla is, of course, a much more mature field at the present time. You've also heard about the fact that you can visualize veins, and you've seen some beautiful results that Dr. Heike showed. This is from my effort at 9.4 Tesla in the human brain from my lab when I was in Tübingen at the Max Planck Institute. This is a comparison between 3 Tesla and 9.4 Tesla, looking at same subject and identical resolution and projection thicknesses, and as expected, you can visualize many more smaller veins at the high magnetic field because of the larger contrast and higher signal-to-noise ratio. Here's another example looking at 9.4 versus 3 Tesla, and you can even use techniques like SWI that Dr. Heike had mentioned, that he had pioneered, to see at these high resolution even intracortical veins, and or phase imaging that he has also mentioned. And you can see that in the human brain now, these are not cadaver brains, these are live human brain images. You can start to see these intracortical veins at these high fields, at these high resolutions. Here's another example of that. And of course, at these high fields, you can see many other structures that are very much of interest to us, such as you can visualize the projections, myelinated projections into the primary, the nerve bundles directly as well. That is another interesting topic. I won't go into it in this talk, but there seems to be susceptibility differences between highly myelinated structures, and you can see these structures, namely the nerve bundles, directly in the high field images as well. With that, I'd like to end my talk. Thank you very much.
Okay, do we have any questions? Okay, I'll ask. I have one. So I was very impressed by the work showing the columnar level regulation of cerebral blood flow. Can you discuss specifically what some of the postulated mechanisms for that are? How it's achieved at such a spatially fine level?
The blood flow mechanisms for blood flow regulation is still actually is a debated topic. And blood flow regulation, for example, even very large vessels seem to be regulated even by direct neuronal control. So it's my view of it is essentially you have regulation at very distal big arteries that are supplying that area. You have, we've known to that there is regulation at the arterial level, and about the same time when this work was done, there were some very detailed structures of blood vessels, images of blood vessels that emerged, where you can actually see structures around capillaries that can, I forgot the name of these structures at the present time, but that can act as potentially regulators that are, that can actually constrict those vessels as well. So there does seem to be some structures in the capillaries that allow that flow to be regulated at that, but exactly what the mechanism to trigger that, this is still unknown.
Okay, thank you. Well, let's thank our speaker again, and I think afterward we'll have a announcement from our...
Thank you very much, Camille. I was just asking if he could send me one of the 70 scanners. It would be very nice. A few announcements. First, breakfast, as people have been not getting up at 7 in the morning. The breakfast is going to start at 8 o'clock tomorrow. And also, we need to have Jesper Hegemeyer and Robert Zavadanoff for their presentations in the next session. Please check with Emil. We're going to take 15 minutes. And please get your coffee, chat a little bit, but please bring your snacks and coffee back in here so that we can get back on time for the next talks. And those people who are talking, if you can talk 5 minutes less than your time so we have time for questions, that would really be ideal. Thank you, and see you in 15 minutes.